I'm very pleased to introduce Professor Kim Mutsert. Kim is originally from the Netherlands, but she moved to Louisiana about 20 years ago to do a PhD with the one and only Jim Cowan at LSU in Baton Rouge. And let me add that she had had connections to UBC for most of the time she's been in North America, making it extra pertinent that we hear from her today. Carl Walters was a friend of Cohen, and he told me about the impressive field work on marsh fish, area fish populations that Kim did for her PhD, and how they were discussing how to model this spatially. And I've known Kim since around the time she finished her PhD. That's when I joined the supervisory committee of Christy Lewis, also at LSU, that was following up on Kim's work. And I saw then the impressive work they were doing in Cowan's lab. It was really inspiring. Kim then moved on from LSU to a faculty position at George Mason University in Virginia, that's close to Washington, D.C., where she became the associate director of the Potomac Center. Next, they moved to University of Southern Mississippi. That's where we hear from, where she's an associate professor based in beautiful Ocean Springs. Over the years, Kim has developed to become a leading expert on coastal and estuarine fish ecology. And she and her lab works with a combination of field monitoring, lab experiments, and ecosystem modeling. And I really want to emphasize the modeling. After all, that's how you make sense of the data you get from monitoring and experimental work. And in Kim's case, take it to the room where it happens, where decisions are made. And let me illustrate this. Irrigation and dams have caused the sediment loading at the Mississippi River mouth to decrease from 400 to 150 million tons per year, so to less than half. And that has big impacts on land loss in the river delta. So Kim has worked with US authorities to evaluate the ecological impacts of alternative river diversions to counter the loss. And that work has direct consequences for both people and nature. And I can assure you that the work has to stand up to scrutiny when it's for environmental impact assessment, where the stake of a decision is in the hundreds of millions of dollars. So I've seen how Kim has developed to become one of the leading spatial ecosystem modelers in the world. Her work is inspiring and cutting edge, as we'll hear. She's developing new approaches for coupling spatial models and evaluating the results among mud cells. She also takes care to guide fellow scientists, and her extensive publication record testifies to that. And notably, I'd like to mention the work she led on a recent Mammoth chapter on spatial modeling. It will stand for years as the key reference. And I've had the pleasure to work with Kim on model development, many publications, and organizing a couple of international conferences. And I really admire the skills and education she brings. And with that said, I'm very happy to welcome Kim to give a talk called Evaluating Response of Fish and Shellfish to Anthropogenic Impact Mitigation in the Mississippi River Delta and Coastal Region using coupled modeling approach. Only wish that it had been in person, because I really miss you, Kim. But welcome.
Thank you so much, Willy. I'm really honored to be invited here for the seminar. Let me share my screen. All right, well, thanks for that great introduction. I would actually also like to start with a land acknowledgement of my own. I am located in Mississippi, and I acknowledge the original inhabitants of the land upon which the Hattiesburg and Gulf Park campuses are built. I honor the unnamed ancestors who were the first to walk the Pine Hills and Gulf Coast Plains, and those whose origin and migration stories, medicine, and plant knowledge are of this place. I offer my respect to the living descendants of those people and to those still occupy this territory as a sovereign nation, the Mississippi Band of Choctaw Indians. So what I would like to talk to you about today is how fish and shellfish respond to coastal restoration efforts. And I'll do that by providing two case studies. One looking at mitigating wetland loss in the lower Mississippi River Delta. And this is a goal of the Louisiana's Coastal Protection and Restoration Authority. And the other one is reducing the size of the Northern Gulf of Mexico hypoxic zone, which is a goal of the Mississippi River Gulf of Mexico Watershed Nutrient Task Force that you can abbreviate as the Hypoxia Task Force. So restoring coastal areas, that sounds like something fish and fishers would be on board with. So what's not to like? These can only be positive effects, you can think. But there are some things to consider. So, for example, reducing hypoxia can indeed be expected to benefit fish and fisheries, but the nutrient load reduction to meet those hypoxia reduction goals could reduce productivity. And these coastal restoration and protection projects designed to mitigate wetland loss, you can imagine that they can benefit fish and fisheries because you protect their habitat. But when those projects are implemented, there could be effects to the fish and shellfish there. They can be displaced. It may change so that it may be not the habitat they're used to and like to live in. So natural resource managers need to make trade-offs. That's hard enough, but they need the information to do so. So these trade-offs need to be informed by science. So my methods to look into this have been using EcoPath with EcoSim software, and I'm going to assume that people listening here are very familiar with this, with Villy and Carl's work on it. So long-term simulations of a future with and without these restoration actions, and also different versions of these restoration actions, are compared to provide insights into relative effects on fish and shellfish. So relative here means relative to not doing it. And ecospace models were used to do this, so developed in EcoPath with EcoSim, and then coupled to other models that give the input, so either landscape or water quality models that can simulate how the amount of wetlands change, how much the water quality parameters change over that time, and so on. So, as I said, I'm assuming you're familiar with EcoPath with EcoSim, but perhaps more in a sense of a food web model, which can be used to look at different fisheries management scenarios. So what makes EcoPath with EcoSim suitable to simulate the response of fish and shellfish to changes in the environment? And Villy mentioned my dissertation in Louisiana a bit, and I'm going to start with that. For my dissertation, I looked at the effects of river inflow through a diversion that you can see here, indicated with this arrow, into the Breton Sound, which is this estuary here. And this diversion creates this salinity gradient. We were interested in how that affects fish and shellfish communities in this area, and also how the information gained from this could be used to, as a proxy really, to larger diversions that are planned in this area. So you can see this 1, 2, 3 here. I used the salinity data from those 3 areas as scenarios in EcoSim. So I built an EcoPath model of the estuary and then used the salinity from those 3 areas as 3 different, like a low salinity, medium salinity, and high salinity scenario. So this could be done with forcing functions in EcoSim. But what wasn't there yet was having a species-specific response of each of the different species in the model to the salinity. So for me to look at how salinity would change the community composition, that needed to be included. So I communicated this to my advisor, Jim Cowan, and he talked to Carl and asked if that would be possible. So Carl said, absolutely not. And subsequently proceeded to make it possible. So both he and Villy worked on this. Villy created a new version of the model that was sent back to me. And basically with that new capability of how EcoSim can simulate the nekton response to salinity changes. So what I did is I used these field data to create these response curves. And you can see the examples shown here are largemouth bass, a fish that prefers lower salinities, a spotted sea trout, a fish that prefers higher salinities. So they respond differently to salinities. So it makes a difference that each of these species have their own response curves in the model when we include these forcing functions. So at the time this was included in the group info spreadsheet in EcoSim, these curves were translated into an optimum and a spread, basically a standard deviation from that optimum that I could type in. So this was very useful and really got an upgrade pretty soon that's applied to EcoSpace as well. I was doing this in EcoSim, and that is the habitat capacity model. So with that, all these different types of curves, you can recognize perhaps the salinity curve here, but a relationship of fish with an environmental function can take all kinds of different shapes, as you can imagine. So, for example, this first one, maybe dissolved oxygen, which is another topic I'll be talking about in addition to salinity. So with this habitat capacity model, I could now include various environmental preference functions, more than one forcing functions, which all together determine the habitat capacity of each cell in an EcoSpace model. So basically that was taken care of. But then I ran into a new problem going from EcoSim to EcoSpace. I realized these environmental variables and stressors, such as salinity and dissolved oxygen, vary in space and time. So this is the exact same data that you just saw in 3 lines that I used as scenarios in EcoSim, but now in space and time. And this is, instead of just including a line or including a map, I wanted to include a spatial and temporal dynamic forcing function. So this was actually possible in the 5th version of EcoPath, but it disappeared from the 6th one. And Carl, who was at this point my co-advisor for my postdoc, had ran into the same issue and said, oh, I have a plugin for that, which he called the Phlegm Reader, which he used in Apalachicola Bay. To look at very similar problems, salinity, stress on oysters. And I was able to obtain that plugin and add it to my model version. And I now could read in salinity and hypoxia in each time step in each cell of my model. Now, this also got an upgrade with the spatial temporal data framework that is now a very useful portion of EcoSpace, where you can read in basically a new map of environmental information of each of the environmental parameters that you want to include into EcoSpace with each time step. So now that we're all set model-wise, I started using these tools in my models. So again, we're in Louisiana, the Louisiana coastal area indicated in this map right here. And this area, this is zoomed in. What you're looking at here is the Mississippi River. You may recognize the Bretton Sound that I just presented as part of that lower Mississippi River Delta. This entire area is affected by high rates of wetland loss. So the loss between 1932 and 2016 was measured to be close to 5,000 kilometers square, and this loss is continuing. So to mitigate this decline, there's a large coastal master plan in effect that's adapted every 5 years that includes a suite of protection and restoration projects that are meant to, mostly meant to mitigate wetland loss. So in order to pick which projects should be part of this plan, a lot of landscape models were used, and I was involved in this by adding an ecospace model that can then look at the effects of each of these combinations of projects in the Mississippi River Delta. So what I'm presenting today is eventually a suite of projects were chosen to look at what are then the effects when we implement all these projects that are written into the Coastal Master Plan. What are the effects on the biomass and distribution of fish and mostly fishery species of implementing that plan versus a future without action, so just not implementing any of these projects in the estuaries over a time period of 50 years. So the modeling framework was indeed a coupled modeling framework. The Coastal Master Plan is, these projects are simulated with what's called the ICM, the Integrated Compartment Model. These are mostly a suite of landscape models. They have various outputs, though, that can affect fish and fisheries, such as salinity, temperature, total suspended sediments, total chaldal nitrogen as a measure for productivity, and marsh coverage. So all these outputs were read into ecospace using the spatial temporal data framework at different time steps. So most, I would say, were on monthly time steps. Salinity, temperature, total chaldal nitrogen influence each of the species in the model. The wetland marsh, the amount of wetland build or loss, was updated annually. And we've realized that certain species, the temporal resolution of a month just wasn't fine enough to get a correct response. So we included, I would say, a subroutine that looked at the daily habitat suitability. So these were habitat suitability models of oysters. So those were sensitive to smaller changes on a smaller timescales than month, of salinity, temperature, and total suspended solids. Those were then transformed into these, what we called oyster environmental capacity layers, which are really habitat suitability models that were then read in on a monthly time step into ecospace. And then we ran ecospace over 50 years, again, with these projects implemented compared to not implemented. So real quick then, indeed, an ecopath model was developed here again. I had 55 groups in the model. Then in EcoSim, I used EcoSim for calibration. So that's just the temporal dynamic simulations, but the modeling framework was an ecospace model. This is a spatial domain of the model. What I'll be talking about today is just that lower Mississippi River Delta, but in the regions within that delta that are of interest to managers are the lower Barataria Basin, Breton Sound, the lower Pontchartrain Basin, and the Birchfoot Delta. And the resolution of this model is a 1 kilometer size grid cells. The scenarios modeled, again, were the biomass and distribution of fish and shellfish in response to implementing the projects that are accepted in the 2017 Coastal Master Plan over 50 years, and then a future without action. These were run under varying climate conditions. The lowest projected sea level rise and other things were varied in there. was 0.43 meters over 50 years. The highest projected sea level rise was 0.83 meters over 50 years. There were 55 groups in there, but there was a focus on economically important species. Blue crab, black drum, you see them up here, brown shrimp, Gulf menhaden, largemouth bass, eastern oyster, spotted sea trout, and white shrimp. What you see here is again that same lower Mississippi River Delta and the regions in that were transferred into my model, and the projects that were implemented over that time. These projects have different start times over these 15 years. In the model simulations, those start times were adhered to. What shows up as small blue lines are the biggest projects. Those are large sediment diversions that are basically a much larger diversion as I was studying for my dissertation, that Carnarvon diversion that led in Mississippi River water back into these estuaries to rebuild the wetlands there. So basically the Mississippi River is levied off by true controlled opening, letting that water back in. Nutrients and sediments are reintroduced into the estuaries, and that helps mitigate that wetland loss. Those aren't the only projects in there, though. There's various structural projects as well, such as marsh creation and different ridges that are all implemented. And whichever effect they have on marsh area, salinity, nutrients are all transferred into the ecospace model. So here are the results. What you're looking at here are box plots where CMP is where the projects are all implemented. This is year 50. And then the future without action is in green. Each of these plots are the different species that I just mentioned. And you can see the biomass on the y-axis, and these different box plot comparisons are the different regions. So the Birdspoot Delta, Breton Sound, Lower Barataria, and Lower Pontchartrain. Now, what you can already kind of generally gather from these plots is first, the differences in biomass are not that different. And oftentimes, the implementation of the plan is the biomass ends up being higher than a future without action. In some cases, there is indeed a little bit more biomass in a future without action than in a coastal master plan future, but none of these differences are actually significant. A very interesting result is eastern oyster. There's a lot of pushback to these large sediment diversions because the fresh water introduced through those diversions is expected to have a negative effect on the oyster population. So there's pushback to the extent of lawsuits of not having this happen. So I'll do a little bit of a deeper dive into eastern oyster. How is it that actually the coastal master plan implementation in these model simulations do not show reduced biomass of oysters? So here we see it spatially distributed. So there's year 0 where these oyster banks are shown, and they're in year 50. Now, what is true, and probably one of the main reasons for these results, is that in the future without action, there's expected to be less oysters altogether. But compared with a coastal master plan implementation, there's a lot less loss of these oysters. And interestingly, there's a lot less loss under high sea level rise. So we're also facing climate change, which is included in these models. So to have a little bit of a closer look into that, there's biomass reductions in the future without action. With the coastal master plan, there is inflow of fresh water, and nutrients, which allows for a lot of phytoplankton growth, which feeds these oysters. But also, while there's fresh water moving into these estuaries, the sea level rise actually causes saltwater intrusion at the south side of these estuaries. The combination of that freshwater inflow and saltwater intrusion actually keeps an area suitable for oysters at that southern end of the estuaries, maintaining that habitat. While even though it is there right now, under that saltwater intrusion scenario without the projects, that would actually disappear, making this difference positive for the implementation of the plan. So if we look at that difference between the low and high climate change scenario now, where high is more sea level rise, we can see, first off, overall, that's why I put this red line in here, is that this is the biomass of the Coastal Master Plan implementation compared to a future without action. Any of these values ending up above 0 means there's a positive difference of implementing the plan. And in some cases, and this varies per species, there is a slightly higher biomass expected under high sea level rise. And that is because of that combination of freshwater inflow that kind of counteracts saltwater intrusion. So overall, the effects are not as negative as a lot of people expect that have that fish for oysters, shrimp, and so on. But there's going to be a change. So coastal areas need to prepare for a changing environment. So a main take-home message is that a future without action is not a future without change. So without action, there's going to be wetland loss. And coastal restoration and risk reduction projects are part of the strategy to counter that. So there will undoubtedly be effect on living resources in these areas as they are right now, and definitely a change in distribution could have a lot of effects enough for fishers. They may need to go somewhere else. But the simulations such as the ones conducted show that there are not large negative impacts on the living resources. Again, insights are useful here as well into how the distribution changes. So there may still be indirect impacts on fishers if they do not know where to set the spat of oysters. So a lot of information needs to accompany these changes to make the transition possible. All right, and I wanted to add this other topic that I've been working on, really in that same area, but now a little bit more offshore, where a large area of bottom hypoxia forms each year. So it's fueled by the nutrient loading from the entire Mississippi River watershed, but it all comes out in this area right here. So it's been well known that this area forms every year. It's a summer occurrence, so it's a spatial, temporal stressor. But there's less knowledge on how this affects the fish and shellfish in this area. And important players here are red snapper, white shrimp, Gulf menhaden, and Atlantic croaker. And specifically in what the effects are of reducing this hypoxic zone. And for this, I worked with the Hypoxia task force that have a goal of reducing the 5-year running average of the Gulf hypoxic zone to 5,000 kilometer squares. And what you can see in this graph, where area is on the y-axis and time is on the x-axis, that goal is something that is not currently happening. So right now, this is already a little bit old information, but the 5-year average is a lot higher than that goal of 5,000 kilometer squares. So again, my involvement here is what the effects are on the living resources of the reduction of nutrient loading that would need to happen to get at this goal. So they also have an interim target of reducing the load of nitrogen and phosphorus into the Gulf of Mexico by 20%. So we look at what are the effects of reducing nutrients by 20%, but also to what amount of reduction is necessary to reach that goal of 5,000 kilometers squared. So this was done with another team as well at Dalhousie University. Katja Fennel and Arnaud Laurent are the people I worked with on this. Their model showed that this can be achieved, this goal, by reducing nitrogen and phosphorus loading between 40 and 50%. So those are the scenarios we ran as well. This was a co-produced project with various workshops, and the information that we learned from these workshops were to start with what species to include in the models, what scenarios to run, but also what type of output metrics and data visualization are useful for the managers to actually use this information. Also, that we needed to include an uncertainty analysis before output could be accepted. And while we did include training and how to use these models, there was less interest in that. So we concluded from that the decision support tool that visualizes the output is preferred rather than having people run their own models. And then what species to include in a decision support tool. So even though there's more species in the model, that may not be what needs to be included in decision support tools. So here again, I used EcoPath with EcoSim. There are 66 groups in this model. All of them had juvenile and adult life stages or multiple stages, depending on the ontogenetic shifts that occur within each species. The ecospace were run in IBM mode. What that does is that the species are in groups of packets really that move over the model area, which does a much better job at looking at the kind of localized mortality rates based on these spatial temporal stressors that are thrown at the species in the model. And we ran spatial Monte Carlo runs to get at this uncertainty analysis. This is the EcoPath model that I developed. So there's fishing in the model as well, all these different species. And you can see with this network that there's a lot of connectivity in this Northern Gulf of Mexico food web. And what you see on the y-axis is the trophic level of the different groups and the colors. The environmental response curves were included. For this, I could use a lot of data again. This is CMAP surveys. They do trolls. The catch versus bottom DO was used to look at what the different responses was of all the different groups in the model to dissolved oxygen. This is the model area used. What you can see in this orange is that I had to exclude some areas because they weren't receiving, those cells weren't receiving output from the model that it was coupled to. And we only want model cells active that get dissolved oxygen and actually nutrient information from the coupled lower trophic model. So here you see a clip of output from that lower trophic model. So it's a ROMS-based physical biological model that also outputs chlorophyll and actually even phytoplankton as well. Kind of the top group there is zooplankton. And what you're looking at here is a run through 1 year. So the example year is 2001. You can see that the chlorophyll A at the top is kind of growing from winter into spring and summer. And once that phytoplankton dies and sinks, the bacterial decomposition at the bottom takes out the oxygen, and that really only forms in summer. So these red colorations are the oxygen levels at the bottom. So you see that forming, but you also see that dissipating again. So this problem is truly a problem in space and a problem in time that we want to simulate with these spatial temporal model runs. And here you see an example of species response in space. So we first wanted to see, like, is there even a response of our species, right? I'm now running through the months of a year. This is 2008, where there was severe hypoxia, and you see it happening, right? The shrimp clear out through movement, but also through mortality of the areas where there's hypoxia, but they also return back. So from all these runs, I mentioned the species of interest: red snapper, Gulf menhaden, Atlantic croaker, and white shrimp. We looked at all the output of that. What you're looking at here is at the end of the run in March and in August, while there is no nutrient reduction. So this is under a hypoxia or conditions where no action is taken, basically. So you can see in August, in summer, that there is an effect of that hypoxic zone. So we do know that reducing that hypoxia does benefit these species, that they are affected by it. And you can see that the effect is less in March. You still see some traces of it, so that hypoxic zone comes back every year. But there's definitely, it's not just full mortality, there's definitely movement as well. So when we reduce the hypoxia, so now we see the no reduction scenario in that first column. Now the species are kind of in rows: Atlantic croaker, Gulf menhaden, red snapper, white shrimp. This is August at the end of the model run, so that represents 2035. And then each of the reduction scenarios: first 20% reduction, 40%, and 50% reduction. So you can see when you look at the distribution that it does matter in a positive way when we reduce hypoxia. So you see, and it differs per species again as well. Atlantic croaker are not as affected by hypoxia. They're pretty resistant. But there is some effect here of the hypoxia that becomes less and less the more you reduce it. And you see that by most species occurring, some more than others. I mentioned the uncertainty analysis. We did spatial Monte Carlo runs and saw kind of what the probability of the outcome of biomass is of each of these runs. This also gave us confidence in the model. If this is not a nice bell curve, that's not great. Or when your base run is somewhere way outside of these probabilities, that's not great either. And then we used basically the standard deviation of these probability density plots to look at kind of a uncertainty band around our output. And I've indicated that here with these dotted lines. What you see here, so that's why I first wanted to show the spatial distribution changes. We know now that it matters, right, reducing hypoxia. But when we look at hypoxia of the total model area, or biomass of the total model area, there's not that much differences in biomass when these nutrients are reduced, and those are the colors here, as compared to the baseline run of the different species here. So we have red snapper, we have Gulf menhaden, we have Atlantic croaker and white shrimp. And oftentimes we see a little bit less biomass of these species under the nutrient reduction scenarios. What this really signals is that the nutrient reductions needed to reduce hypoxia also reduce productivity. And these two, and I've also done runs where I ran either one or the other. I can, artificially exclude the hypoxia benefits of nutrient reductions and the other way around, only hypoxia, no nutrient reductions. We do see a negative effects of reducing nutrients, a positive effect of reducing hypoxia. And these two effects cancel each other almost out to get these type of results. So there's various other reasons, of course, to reduce the nutrient loading. But the nutrient loading overall, if we look at total biomass, is not really either negatively or positively affecting the species in those areas. So I showed the spatial distribution to show that, well, the spatial distribution does change, and that does have consequences to fishers, for example, so they may not have to go as far out if the hypoxic zone is reduced. So there's positive effects in different ways, but total biomass really doesn't see a big difference with these nutrient reduction scenarios and hypoxia reduction scenarios. So I quickly wanted to show this decision support tool that we have. Managers can click these different load reductions. Again, these are goals. It's very hard to reach these goals, so it's difficult to just implement a nutrient reduction scenario. It really takes best management practices in the entire watershed to get these nutrients down. So it's not something they can easily turn on and off. But with this, they can look at what are the effects if it does happen. This is also published in a separate paper where we really focus on the code production aspect of this and the development of the decision support tool. So the conclusions of that work is that the distribution is more strongly affected by hypoxia than total biomass of most fishery species. So the positive effects of hypoxia reductions are counteracted by negative effects of reduced nutrients, because that reduces primary production, which reduces secondary production. These spatial shifts are still meaningful to practitioners. There's a very known effect of fleets needing to move to the edges of the outside of the hypoxic zone to fish. So reducing that area can have positive effect to the, for example, the shrimp fishers is a well-known example and published example of fleets congregating on those edges. And the decision support tool really facilitates visualizing these effects of these planned nutrient reductions. All right, so with that, I would like to leave 15 minutes for questions. I would like to thank all my co-authors throughout these years. Again, Carl and Willy were very helpful in this process. Also, the many research and engineers who participated in the Coastal Master Plan analyses. I've used a lot of data. I'm very thankful to the federal and state agencies that have these large monitoring programs, like LEWF in Louisiana and NOAA NIMS. And I'd like to thank my funders, CPRA and NOAA. And with that, I'd like to take any questions.
Okay, thank you very much, Kim. As already appears in the chat box, if you have any questions, you can just type them into the chat box or raise your hand and we'll get to you directly. I'm actually going to take the precedence and start with a question of my own. When you're talking about relatively complex models over a relatively, a decent time interval, is there an ongoing, if you're implementing these, is there an ongoing way of checking the accuracy of the models? Like 5 years down the road or whatever, is that part of the process, or is it just here's our best modeling, leave it to the decision makers?
Right. Yeah. I mean, I would say I thought, for example, with the Coastal Master Plan, that it would be part of the process, right? It's actually well designed in that it's an adaptive management approach where every 5 years that Coastal Master Plan and the models behind it can be revised. But for this, they actually did a new round in 2023 with a complete focus on the land building aspects of the models. So These are controversial topics. There are actual lawsuits against CPRA of people that are afraid that any of the projects implemented in these areas are going to affect where they fish and how much they can fish. So they have actually chosen to remove these models that directly look into it from their adaptive management approach. So the mechanism is there, but the funding agency and really the state decided not to go that route. So hopefully that is revised and these models are taken back up because of course it's possible, right? We already have some projections for the first couple of years. Not only can we test it, we can then improve it. Like that first round, you know, you have deadlines, you need to build a quick model. There's definite room for improvement. Where discovering contaminants coming out of the river, that can affect the fishery species as well, right? When these diversions are open. That's, for example, not in my model. So there's definite points of improvements that I think should be added, but I have to be part of the process to be able to do it.
All right, thank you. Billy, go ahead, please.
Yeah, thanks, Kim. It was a great presentation. One thing I'm wondering about is what, for both case studies, what did the managers say? Was it along the lines of, oh, this is just model output, or this is interesting? What was your reactions?
Yeah, I mean, I think that is varied as well. I think indeed with CPRA, there are a lot of people that are very interested in it. This paper that I presented on looking at the effects on what was actually implemented in the 2017 plans was in collaboration with CPRA, with folks that are really excited and want to have this information as part of the decision process. But I think the overall decision is it's a little bit they're a little bit more fearful of providing that exact output of what we think, because then it becomes a you know, a state output, right, with my model included in it. This is exactly what's going to happen to fish and fisheries, and it makes it perhaps easier to then sue the state if it's not exactly right. So I would say there is individually, there's enthusiasm. As a full organization, there's a lot of caution there as well. When I followed this up for environmental impact assessment studies that were specific for the diversions, and not really like the scientific work that we publish, but really part of the process, I had to go back to EcoPath and really talk about the resilience of the system through network analysis. That could make the ESA, but not the ecospace runs. So there's a bit of a conservative approach with the output overall. So I think that just takes time. But on the other hand, in the planning phases, I think our results are more incorporated. So for example, the work I did specifically looking at the diversions was at a time when they had still plans of including 4 different large sediment diversions. And our results were in combination with results of other models, helped with the decisions to not go forward with the 2 lowest diversions and only with the mid-Breton and mid-Barataria diversion. So in the planning phases, I think there's a lot more uptake. And then truly with the, the ecological impact assessment, they're a little bit more conservative and want to step back and not use this to make what's then seen as predictions that can then be tested. And if they're not true, you can sue the states a lot easier. So that's a long way of saying that the response is mixed, and the uptake kind of varies of in which stage of the process you are. Are you in the part that they can use, to sue the court, sue the state, or are you more into the kind of scientific evaluation of different options, and then the uptake is more positive? Yeah, I think, well, actually, if I'm allowed to talk longer.
Sure.
That was mostly about the wetland mitigation project. So with the Hypoxia project, I think the main issue there is that they have a hard time reaching their goals. So they have goals, but other than the Clean Water Act, there's not a lot of, I would say, legal teeth to achieving their goals. So they can really suggest and help with implementation of best management practices in the watershed. So the Hypoxia Task Force is state and federal agencies throughout the watershed, but it's harder to make it a mandate. Like we need the reduction needs to be this, and so it's so with that in mind, the scenarios that we run are a little bit wishful, and therefore the implementation is lagging behind a bit because it's hard to actually reach those goals right now.
Okay, thanks. Carl, do you want to go ahead with your question?
Yeah, Kim, I'm just endlessly amazed at you. Year after year after year, I keep telling you that you can't do what you do, and yet you do it. Billy and I are so proud of you. I wanted to say that in general, but also a really critical part of, and it answers some of the questions like that David brought up, is that you're not just running a great big model and saying, here's our results, you take them or leave them or whatever. You are encapsulating them into much simpler qualitative statements about consequence. Like saying, well, yeah, if you get rid of nutrient loading, you will get rid of hypoxia, but you'll also lower productivity in those systems. And you got to be really, really careful that that impact isn't much larger. Now that kind of basic qualitative conclusion is very robust to the model details. It doesn't matter whether you're running a giant grid like you did to impress people or doing something more simple. So that I think it is something that people that do these models don't often do. And people who get engaged in adaptive management processes don't often do. They say, okay, here you got a model and you got to use it and you got to add it and learn from it and so on. No, the most of the learning, as you just said, comes from the qualitative things that happen early on when you're trying to figure out exactly what the problem is, as you just said. Good on you, girl.
Thank you.
Thanks, Carl.